Hello, everyone, and warm welcome to all of you who have joined us here today. My name is Bruna Lousada, and I'm the European Manager of Teledyne Hansen, and I'm delighted to be hosting this webinar with Dr. Hanan Osman-Ponchet, the founder and CEO of PKDERM. Some of you may remember Hanan from our last webinar in November, In Vitro Permeation Testing, application to cosmetic products. And during the Q&A session, there were many questions related to the complex process of conducting an IVPT study. So it has taken us longer than expected, but we have worked hard to prepare this very special webinar for you, which is Mastering In Vitro Permeation Testing, a comprehensive guide to conducting an IVPT study, where we will be addressing most of these questions. So today we'll dive deeper into the topic and cover everything you need to know about conducting an IVPT study, including videos and expert tips, and we hope that you will find it both informative and valuable. So just before we begin, I would like to remind you all that all attendees are currently on mute, but you can submit your questions at any time using the Q&A section at the bottom of your screen. We will do our best to address all of your questions at the conclusion of the presentation, but if we run out of time, we will follow up with you directly via email. If you experience any issues with your audio or connection, we recommend logging off and logging back on. And additionally, this webinar will be available for replay on the Teledyne Hansen YouTube channel in the coming days. So without any further ado, I would like to invite Dr. Hanan to take over the presentation.
Hello, everyone. I am very happy to present the webinar on how to perform IVPT. I hope this webinar will be useful for the audience. My name is Hanan Osman-Ponchet, and today I will show you the different steps of conducting IVPT using excised human skin. You will see during this webinar how to prepare skin samples, how to prepare diffusion cells, and you can see how to make topical application, and finally, and this is the most important step, how to separate stratum corneum, epidermis and dermis at the end of treatment period. So let's go. So IVPT, or in vitro permeation testing, is a critical tool for understanding drug delivery into the various layers of the skin, and it plays an important role in the optimization, comparison, and selection process of drug formulation. IVPT allows to study the permeation of drugs through skin samples in a controlled laboratory setting and provides insight into how different formulations, delivery systems, and skin types impact drug delivery. IVPT can also be used for bioequivalence studies of topical generics as an EMA and FDA-approved biowaiver alternative to costly clinical studies. In addition to its use in drug development, IVPT is also used for safety assessment of chemicals, agrochemicals, and cosmetic products. What is the objectives of in vitro permeation testing? In fact, as I said, IVPT is a valuable tool for characterization of the rate and extent of transdermal or topical delivery of test items. The main purpose of this testing is to evaluate how the test item is distributed into the skin, including the stratum corneum, epidermis, and dermis. IVPT is also used to assess whether the test article will penetrate the skin and reach the systemic circulation, and to what extent. This is particularly important because drugs that are intended to be delivered transdermally, as the skin acts as a barrier to prevent the entry of foreign substances into the body. So by measuring the drug's permeation rate and extent, researchers can determine how the drug is distributed in the skin, as well as its potential for systemic absorption. Now, IVPT is typically conducted using a France diffusion cell. France diffusion cell consists on two chambers separated by a biological membrane like skin. The donor chamber contains the drug formulation to be tested, and the receptor chamber contains a suitable medium to simulate the biological environment. The skin sample is placed between the donor and the receptor chamber. In the field of in vitro permeation testing, regulatory guidance documents play an important role in ensuring that testing is conducted according to best practices and is consistent with regulatory requirements. Here we have a list of the key regulatory guidance documents used in IVPT studies. This includes the EMA, the USP guideline, OECD, the Scientific Committee on Consumer Safety guideline, and the World Health Organization for dermal absorption.
Regarding human skin samples, it comes from aesthetic surgery from the abdomen area. You can see how skin sample looks like. On this sample, we already removed the fat corresponding to the hypodermis layer, so that we can clearly see the dermis beneath the epidermis. You have to check carefully the skin samples to see if there is any damage or lesion on the skin that may affect the results. From skin sample, we prepare discs of about 3 or 4 centimeters of diameter. As you know, skin thickness can affect dermal absorption. So we need to measure the thickness of each skin sample in order to ensure an homogeneous distribution of the samples within the experiment. At PKDERM lab, we use an electronic caliper to measure thickness of skin samples. That should not exceed 1 millimeter. So this is why we normally use split thickness or dermatomed skin when we perform IVPT. After the filling of a receptor chamber with the receptor fluid, we place a skin sample on the diffusion cell. And this is done in such a way the dermis is in contact with the receptor liquid, while the epidermis is left at the air surface. We then place the diffusion cells on their support under gentle shaking and at 37 degrees. Transepidermal water loss, or TWL, is a quality indicator of skin barrier integrity. So, when performing an IVPT, we need to check the barrier integrity for each skin sample. At PKDERM, we use a theometer system to measure the TWL. Normally, value of TWL should not exceed 15 grams per square meter and per hour, and skin samples having higher values of TWL should not be included in the experiment. For topical application, different forms of finished product can be used in IVPT. For example, we can use a solution, cream, ointment, or gel. For semi-solid formulation, we use a positive displacement pipette for topical application. The amount of formulation we applied on skin surface should be accurately measured. This is why we weight the pipette containing the formulation before and after application on the skin. By this way, we can know the exact amount of the product we applied on the top of the skin. And the amount applied is used to calculate the absorbed dose at the end of the experiment. After the application of the formulation, the diffusion cells are left for the duration of the treatment period, usually 24 hours. For And here you can see how we collect skin sample from the Teledin Hanson diffusion cell. So you can see clearly the treated area of the skin where we can see the applied formulation on the top of skin surface. And now I'm going to show you how we can remove excess formulation. And this is really the most important step to do in order to get very accurate results. So for that, we use 5 cotton swabs to remove excess formulation. and then we use 2 tape strips in order to be sure we completely remove the excess formulation. After removing excess formulation, we can separate the different skin compartments. We first separate the stratum corneum by tape stripping. So how we perform tape stripping? So as you can see, we press an adhesive tape onto the surface of the skin, and then we remove it abruptly. So this procedure of application and removal of adhesive tape is repeated 10 times. We collect each tape strip in one tube containing an appropriate solvent in order to extract our test article from the adhesive tape. And then we can analyze the compound in each layer of stratum corneum. Normally, one adhesive tape can remove one layer of the stratum corneum. The tubes containing the stratum corneum are then mixed very well and left to under checking for at least 4 hours in order to extract the test compound from the stratum corneum before analysis. After removing the stratum corneum, we can collect the epidermis. For that, we use scalpel blade to remove the epidermis layer, as you can see. The epidermis is then collected in fast prep lysing matrix tubes from MP Biomedicals. To collect the dermis, we use a biopsy punch in order to collect the treated area of the dermis. And then the biopsy containing the dermis is cut into several small pieces using scissors and tweezers. Pieces of dermis are collected in fast prep lysing matrix tubes containing ceramic grinding spheres. These spheres helps to grind hard tissues like the dermis and epidermis. After collection of epidermis and dermis samples, we need to homogenize them for better extraction of the test compound. To do that, we use the fast prep instrument in order to grind epidermis and dermis samples. And at the end, we centrifuge the samples and the supernatant containing normally the test compound is collected. So at the end of the experiment, we get several samples. Samples corresponding to the receptor liquid, samples from the stratum corneum, from epidermis, and the dermis. The samples are stored at minus 20 degrees Celsius before being analyzed.
So IVPT samples need to be analyzed to determine drug permeation and assess formulation performance. One common analytical method used for IVPT sample analysis is LC-MS-MS. So why LC-MS-MS? In fact, because it is a highly sensitive and specific analytical method that can detect low levels of drug molecules in samples. The limit of quantification for analysis of IVPT samples is typically very low, almost 1 nanogram per ml, making LC-MS-MS ideal for detecting even very small amounts of drug molecules that may have penetrated the skin. So high sensitivity is critical for accurate and reliable measurement of drug permeation in IVPT studies. Other analytical methods may also be used for IVPT sample analysis, but LC-MS-MS is often preferred due to its high sensitivity and selectivity. So in conclusion, we saw that IVPT is a critical tool for assessing drug permeation and formulation performance. To obtain accurate and reliable IVPT data, proper sample preparation and analysis are essential. This includes quality control of skin samples, careful application of formulation, removal of excess formulation, and separation of each skin compartment. So IVPT studies are useful in bioequivalence studies, helping to establish the bioequivalence of generic formulations to their branded counterparts. Additionally, IVPT can help guide formulation development and optimize drug delivery, ultimately improving patient outcomes. So overall, IVPT is a valuable tool for understanding drug permeation and has important implications for drug development and patient care.
Thank you so much, Hannah, for this excellent presentation. I'm very confident that our audience found it informative. So we will now move on to the Q&A session, and I would like to remind everyone that you can submit your questions using the Q&A button located at the bottom of your screen.
So thank you very much for the audience, and I would like to thank my amazing team for their big job doing this presentation and performing IVPT studies in the lab. Thank you very much.
Thank you. Hey, Hannah. So we have a lot of questions from our audience. The first question for you, can we get 100% of the active from the cream? And what is the expected time to get high results?
In fact, it is very difficult to get 100% of the active from the cream because, as you know, the skin barrier is very effective, and its role is to avoid any penetration of xenobiotic or any compound. What we expect normally is between 1% of the applied dose that can reach skin, and also it depends on the active ingredient, on the molecular weight of the active, and also on the formulation, because some formulation can increase or enhance dermal absorption, and some other can slower dermal absorption. So it depends really on the formulation, on the active, but we never reach 100%. Regarding the expected time to get high results, in fact, in the in vitro IVPT, we do the experiment or the application time for 24 hours sometimes, but it's very rare, and I could not recommend that we treat for 48 hours. So we cannot do more, but we can translate and extrapolate the result we get in vitro to the in vivo situation.
Very good. Thank you, Hannah. Next question is about the transepidermal water loss value that you've mentioned in the presentation. So was that based on your experiments or is in the literature?
No, in fact, this value has been published in the last draft guidance of the FDA in 2022. They mentioned this criteria of 15 grams per square meter per hour as acceptance criteria for TWL.
Very good. Next question, is the skin sterilized before testing, and have you experienced any issues with contamination during the tests?
We don't sterilize the skin before testing, but what we do is to disinfect briefly with ethanol solution. And we work properly. We work mostly in PSM, in safety cabinet, to avoid any contamination. And we never experienced any infection or contamination during our experiment.
Perfect. Next question is about sink conditions. So how do you check sink conditions?
Yeah, it is absolutely necessary to check the sink condition before doing the IVPT. I didn't mention these conditions in the webinar, but in the first webinar, I think I mentioned that you should check for sink condition. You should check for the solubility of your active in the receptor fluid and also the stability. So there is preliminary experiment to do before doing the IVPT in order to check for the sink condition.
A question about cosmetics, so probably someone that attended our last webinar. So for using cosmetics, would you still take the receptor fluid measurements, considering that cosmetics are not supposed to reach the serum in the skin? And what is the significance of a cosmetic product reaching the receptor fluid?
Yeah, in fact, when you do the IVPT for cosmetic products, yes, so we continue to measure the concentration of the active in the receptor fluid in order to show that the cosmetic active or the cosmetic ingredient is not absorbed. However, if in the case where the cosmetic active is absorbed in the receptor liquid, it is not really a problem if you have all the data showing that the safety assessment is okay, and if you determine the margin of safety of your active ingredient. But you should absolutely show what is the concentration in your receptor liquid. Of course, a cosmetic product normally designed to be not absorbed, but we have to show that and to demonstrate that.
And I believe that would be for safety as well.
Yeah, absolutely.
Reasons. Okay. Very good. You mentioned the threshold for transepidermal water loss. There's someone asking if you know the threshold for electrical resistance testing, which is also an option for the barrier integrity test. Do you have experience with electrical resistance?
We can use absolutely the TER to measure barrier integrity, but there is no threshold mentioned in any guidance at my knowledge. But for me, it is in our experience we use much more TWL than TER. And normally you should set up your database, your historical data in your lab when you for the measurement either for TER and the TWL. But I can't give any threshold because it depends on your diffusion cell, it depends on your skin sample, it depends on your device you use for the measurement.
Okay, very good. Another question. What is the storage condition for a skin sample during the storage before analysis?
Before analysis, skin samples are stored at -20 degrees Celsius. We normally keep them for a maximum of 1 year. We don't use skin samples after 1 year of conservation at the -20.
Does the tissue do not degrade over 24 hours? And if acid is included to kill microorganisms?
Sorry, could you please repeat the question?
Yeah, does the tissue not degrade over 24 hours? Because you mentioned the test has to be at least 24 hours.
Normally, no, it doesn't degrade. If you use frozen skin samples, normally it is not a viable tissue. What you are looking at, it's just a passive diffusion. So you are not looking at the metabolism activity of your sample, but no, there is no degradation. What you can observe if the experiment lasts more than 24 hours, sometimes we have a separation between the basal layer of epidermis and dermis. And regarding the second part of the question, is that about
if you include anything to kill microorganisms that may grow in probably medium and
some people add some antibiotics in the receptor liquid, but we don't do that in our lab because we avoid any interference for the analytical analysis. But if you work in properly, if everything is clean, normally you don't have you cannot have any problem with contamination.
Thanks, Hannah. Next question. Can you also use IVPT studies to test dosage forms like patches?
Yes, yes. We can use it for patches absolutely. And in this case, we look at the transdermal delivery. Yes, there's many examples published on transdermal delivery using patches in IVPT.
Why do you store samples at -20°C before analysis? And why cannot not assess immediately without storing?
You can absolutely. You can analyze immediately, but if you cannot analyze immediately because it is the planning of a logistic reason, you should store them. And in this case, normally when you set up and validate your analytical method, you look at the stability in the storage conditions. Meaning, if you store your samples for 1 month at -20 before analysis, you should do a stability study for 1 month to show that your active or your compound still stable in these conditions.
Do you have experience with buccal mucosa or buccal tissues? And they're asking if the procedure of the permeation is the same.
No, I don't have experience with buccal mucosa, and I think it is like a 3D model, like a 3D arachide, a constructed human epidermis model. And in this case, we don't use this in transdiffusion cells. Normally, we use them in transwell or insert. Yeah. But I don't have experience in permeability on this model.
Okay. Well, for the person who asked, we have more people maybe working with buccal mucosa, and we can discuss later as well. Well, there's still a lot of questions, but let's see. So what kind of buffer is used for the collection of the stratum corneum, epidermis, and dermis?
In fact, it depends on the molecule and on the compound that we are testing, because normally the compound should be soluble in the solvent we use, and this is defined in the preliminary experiment. So we try different solvent, different buffer, and we choose the best one suitable for the compound. So it depends really on the compound. There is no universal solvent or buffer.
Very good. Okay. What is the current variability observed between cells in replicate experiments?
The variability is in general high in this kind of study. Why? Because the variability of skin sample is high. We have inter-sample variability and inter-donor variability. This is why when we conduct this kind of study, if it is a regulatory study, we have to use at least samples from 6 donors in order to reduce the inter-individual variability. And for each donor, we have to use at least 4 samples. So this is huge. However, when there is in our experiment, we were successful to reduce this variability by using different tips. For example, the removing of formulation excess is very critical, and the way you remove the excess is very critical and may affect, may impact the variability you may have in your study. And also the experience you have when conducting the study. There's small tips, very specific, you can do to lower or to decrease the variability of the study.
Thanks, Hannah. Another question: how do you ensure a good product spreading over the skin with a small amount of product dosed?
Excuse me, sorry, could you please repeat?
Yes, of course. How do you ensure a good product spreading? So how you spread your product over the skin when you have a very small amount of the dosage form, so the product that is being dosed. Do you have any technique?
Normally we use 1 square centimeter of skin, and we apply between 5 and 10 milligrams per square centimeter. So it depends on your formulation. If it is cream or solution, you spread with when you apply with the pipette. So there is no, normally it is not difficult to spread carefully your formulation on top of the skin.
Okay. How can we ensure complete removal of epidermis layer during the skin biopsy?
You can see easily when you remove the epidermis, because the dermis and epidermis are very different when you look at these 2 layers. So when you remove the epidermis, you can be sure that you removed all the epidermis, because there is no keratinocytes that are left on the top of dermis. So it is really something very clear, and you can see it.
There are a few questions about how you pre-treat your samples before HPLC analysis. I know Hannah mentioned in the presentation you're using SDMS, not HPLC. Do you have any experience with HPLC, Hannah?
Yeah, in fact, the LC-MS is a kind of HPLC, but it depends on the detection mode. The HPLC can be used depending on your molecule and on the sensitivity of your method. As I said, we need a very sensitive method, analytical method, in order to detect what is absorbed in the receptor liquid and also in the dermis and skin. So if your HPLC method is sensitive enough to reach a very low detection limit, why not? You can use it. So you have we need to have an analytical method that is sensitive, accurate, reproducible, and selective. So if you have all these parameters okay and your method is validated, you can use HPLC.
Very good. I think this will be our last question before we close our session today. So the samples, there's still a few here, just so you know, so we'll probably get back to everyone via email, but I'll finish with one last question. The samples from IVPT normally contain salts. Can it be injected on LC-MS directly, or it requires some kind of solid phase extraction?
What does contain
salts?
Salt. Yeah. Normally you should extract your samples before injecting in HPLC, of course. You can use solid liquid extraction. Yeah.
Okay. So I think that's the end of our session today. Thank you very much, Hannah, for sharing your knowledge with us today. And of course, to all of you who participated and asked many, many questions, great and insightful questions. As I mentioned, we will, if we didn't, there's more than 50 questions here, but if we didn't manage to reply, we will get back to you via email. So this concludes our webinar today. Just a reminder, the recording of this webinar will be available on our Teledyne Hansen YouTube channel in a few days. We also encourage you to share it with anyone who might benefit from it. And for more information on the webinar or to access on any other Teledyne Hansen product and resources, please visit our website at teledynehansen.com. Connect with us on social media, and we really appreciate your time, and we hope to see you at our next webinar. Thank you very much, and have a great day.
Thank you for the attendance and for the attendee, and for Teledyne Hansen for the organization of this meeting. Thank you.
Thank you very much, Hannah. Thank you. Thanks, everyone.